a few basic steps for protein synthesis. There are two phases in protein synthesis, which is transcription and translation. The first phase is transcription, and it takes place in the nucleus. Here, the DNA is separated or split and is used to make an RNA copy of the DNA. RNA polymerase is an enzyme that connects complementary RNA and DNA strands together. The DNA needs to be split in order to be used as a template for the mRNA, and it needs to be transcribed exactly how the DNA was originally because it's the DNA that has the instructions for building the proteins. The next step is to perfect the messenger RNA strand through something called splicing. During splicing, non-coding segments called introns are removed from the mRNA so that only coding segments called exons are left. Splicing occurs in the nucleus and is an important step in protein synthesis because since RNA is the blueprint for making proteins, any errors from the previous step will be carried over to the next step and can result in non-functional and faulty proteins. It's important to note that splicing errors do not actually count as mutations, since a mutation is defined as a permanent change in the DNA sequence, not the RNA. However, mutations can cause splicing errors. The next step is the second phase in protein synthesis called translation. Translation takes place outside the nucleus, and it's where the mRNA is actually used to make the proteins. The perfected mRNA is sent to the cytoplasm where it attaches to a ribosome, which is made of ribosomal RNA and other proteins. After the mRNA attaches to a ribosome, transfer RNA comes over carrying a single amino acid and leaves it to form a chain. The tRNA reads a base by threes and only leaves its specific amino acid if the base it reads is complementary to the mRNA. For example, if the mRNA codon reads UAA, which is uracil, adenine, and adenine, then the complementary tRNA would read AAU because uracil and adenine are complementary. mRNA is composed of nitrogen bases, which are adenine, cytosine, uracil, and guanine, where cytosine is complementary to guanine. The way proteins are made is by folding a long chain of amino acids held together by peptide bonds. The tRNA will keep making the chain longer until it reads a stop codon from the mRNA, which is a specific combination of bases that tells the tRNA to stop bringing amino acids. After the protein is completed, it folds itself into a specific shape that dictates its use and function. Certain sections of the protein might twist into a spiral or collapse within itself depending on which amino acids it's made of. The type of protein will determine its shape, and there are factors like nitrogen bonding and hydrophilic and hydrophobic amino acids that will either go away from water or stay where they are.